Today, I'm going to talk about a DNA editing technology called CRISPR. What is CRISPR and how does it work? To understand CRISPR, let's dive into a cup of yogurt. In a yogurt-making plant, active bacterial culture such as lactobacillus is used to convert milk into delicious yogurt. However, from time to time, the bacteria is attacked or infected by viruses, known as bacteriophage. These attacks can bring an entire plant to standstill. Not just a yogurt-making plant, but any industry which uses bacteria in a big way, such as paper making, cheese production, so on and so forth. Scientists have found that bacteria can have a natural defense mechanism to such virus attacks, and it is built right into their DNA. DNA stands for deoxyribonucleic acid. These are long strands of specially arranged proteins found inside the nucleus of every living cell. The strands are sequences of the four building blocks of life: adenine, thymine, cytosine, and guanine. The bacteria's defense mechanism that can be built into their DNA is known by the acronym CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. To understand exactly how this works, let's introduce the main actors in our story. Let's start with the bad guy. This is Dr. Virus. Now the good guy. This is Special Agent Cas9 or CRISPR-associated protein 9. When a virus attacks a bacterial cell, it injects its DNA inside the cell. This is when CRISPR comes into the picture. The cell sends out some detectives. They are proteins called CRISPR-associated proteins 1 and 2. They scan the virus's DNA for its quote-unquote fingerprints. The fingerprint is a sequence of nucleobases called spacers. The detectives make a copy of this fingerprint and send it back to special locations on the bacterial DNA called the CRISPR locus. It is basically a place where the DNA files away this fingerprint by sandwiching it between recurring short sequences of bases called repeats. If multiple virus types attack the bacteria, these detectives save a fingerprint of each of them, making a many-layered sandwich. How does the bacteria protect itself when attacked again? The bacteria first sends out its special agent, its very own 007s. These are the Cas9s or CRISPR-associated protein 9. These agents are armed with the fingerprints of the viruses that have attacked in the past. These fingerprints are called guide RNAs. When a virus attacks again, the agents try to match up the fingerprint of the attacker with the fingerprints on their file. When an agent finds a match, it snips the attacker's DNA to disable it. What scientists have done is modify this mechanism to disrupt the expression of specific genes in an organism. They have come up with a way of assembling a modified CRISPR-associated protein 9 armed with a guide RNA that is a fingerprint for specific genes. Scientists are able to insert these agents into the nucleus of any cell, not just bacterial cells. These agents inspect the cell's DNA to find a match for the fingerprint of their assigned gene. When they find a match, they cut the gene sequence out of the DNA. The cell then tries to fix the broken DNA. The errors in doing this cause a mutation in the gene. This procedure is called gene silencing. It is also possible to add a recipe for fixing the disrupted gene, and the procedure is called gene editing. The recipe is the template RNA or instructions for how you want the gene to be modified. This way, scientists are able to achieve the desired gene modification. To conclude, experts think that the discovery of the CRISPR mechanism is a giant leap forward in many fields. My name is Gargi Nandi, and these are some of the resources I used. I hope you enjoyed my video.